Hello everybody, welcome back to another video of biology of cells and tissues. This one hopefully is going to be a little bit shorter. There's not a ton in here. We're going to be talking about the integumentary system, also known as the skin. Of course, as always, leave a like on the video, subscribe if you are not already, and then any questions or comments you have, go ahead and drop those below, and I will get to them as quick as I can. So, looking at our learning objectives, we're going to be looking at functions, regions of the skin, histology, some of the cells, different components of it, as well as some of the other glands and some other structures of the skin. The functions, these make a fantastic test question. Well, which of the following is not a function of the integumentary system? So you have mechanical protection, protection from fluid loss, it's a physical barrier, thermoregulation, and then also a receptor organ. We'll see a little bit of each of these during our presentation. Here's just kind of the skin structure in general. You can see a lot of these different structures. We have some nervous structures. We have epidermis. We have dermis with a couple different layers in there. We have our hair cells. We have sebaceous glands. We have adipose tissue, cutaneous complex, vessels, a whole bunch of stuff. The skin is rather complicated, but we're going to break it down piece by piece. The skin in general, there are two different types of skin. We have thick skin, known as glabrous skin, and then we also have thin or hairy skin. Then there are two distinct regions when we talk about the skin. We have the epidermis and the dermis. There is a kind of a third layer that we call the hypodermis. It's not necessarily a layer of the skin itself. It's more of like a transitional region between the fascia of the rest of our body and the skin. So it's a hypodermis. It's the superficial fascia. Because below our skin, of course, we're going to have also, we can have some adipose tissue, some other stuff in there, and then obviously going to sit on top of fascial layers that then hold together all of our muscles. Okay, we're going to talk about these types of skin first. We have thick, also known as glabrous. It is smooth and non-hairy. This is the palms of the hands, the feet, soles of the feet, the lips, the ears, the places that hair does not grow. In pretty much everywhere else on our body, you can see you grow tiny, tiny little hairs. And it is called thick because it has a thicker epidermis layer for protective purposes. If you feel, the skin that's on your hands and on your feet versus the skin, even like on your scalp and any other part of your body, you can in general tell the skin on the hands, the palms of the hands and the soles of the feet, they're built a little bit tougher. And we're going to see something specific. This thick skin actually has an extra layer compared to the thin skin. It has this stratum lucidum that the thin skin does not have. We also get a thicker protective layer of this stratum corneum, which is that very first layer. We're going to talk about those in just a little bit. Here is the thin skin. This is the hairy skin. It contains the hair follicles, and it's the type of skin that covers a majority of our body. So talking about the epidermis, again, we're going to be looking at the origins. So we have its embryological origin is surface ectoderm. It is made up of stratified squamous keratinized epithelium, and there are 4 or 5 layers from superficial to deep. We have the stratum corneum, the stratum lucidum. That's the one layer that's in the thick layer only. We'll look at that again in a sec. Stratum granulosum, stratum spinosum, and then stratum basal. And all of these, their main function is to form a protective barrier. So, starting superficially, the stratum corneum. This is the most superficial layer, and it contains only dead, flattened, keratinized cells. All of these cells are dead. These are the cells that we're going to be sloughing off or releasing every single day. Like when you You see like dust and stuff. A good portion of dust is skin cells. So that's the cells that are constantly coming off of the very top of our skin. They also contain a high molecular weight, a high molecular weight tonofilament. These are just bundles of keratin. Keratin is what gives it that harder protective quality. So, and it's meant to be durable in the protective outermost layer. Stratum lucidum. This is only found in thick skin. It's a thin, transparent layer containing dead keratinocytes. These are the cells that make keratin. And its job is to add an additional layer of protection in our thick skin. Stratum granulosum has two types of cells. I should say the cells within the stratum granulosum contain two different additional structures. Keratohyalin granules. These are protein-based, non-membranous granules. And then we also have lamellar bodies, which are lipid-filled, membrane-bound granules specific for waterproofing. These granules, their job is to give us the waterproofing in order to prevent water loss as well. And the cells above in the stratum lucidum and the stratum corneum, they are dead cells because of the waterproofing. No fluid and no nutrients and stuff can get up above them, and those cells end up dying. There's some more pictures of that stratum granulosum. Next, we have the stratum spinosum. And these are layers of cuboidal cells. So we transition from squamous to cuboidal that are connected by desmosomes. If we think way, way back to the very beginning of this course, we talked about desmosomes. Desmosomes are these structures. They have these filaments, and their job is to link and hold together adjacent cells. So that's what they do. So they're connected by those desmosomes. They're also called prickle cells because all the desmosomes, you kind of look here, all the desmosomes linking everything, creating that nice tight barrier, makes them look kind of prickly. These also contain tonofilaments, and they provide strength and flexibility to the skin. More pictures of that stratum spinosum. And then finally, we have our stratum basale. This is the deepest layer of the epidermis. This is a single layer of cuboidal, two columnar, so cuboidal or columnar cells. You can see both depending on where you're at. And it rests on top of the basal lamina. This layer is highly mitotic, meaning that it's constantly growing and dividing and producing new cells. Essentially, what happens with our skin is we're literally going to be dividing. Our stratum basale is going to be dividing, creating more cells, and those cells are going to slowly push their way up to the top and to the top. And so with our skin, as it comes off, it's constantly being replaced by new cells. The function of of this stratum basale is to produce the new keratinocytes, the new cells. Okay, that is the epidermis. A little bit deeper, that's the whole top layer. A little bit deeper, we have the dermis. This is embryological origin is mostly mesoderm. It's made up of dense, irregular connective tissue. It has type 1 and elastic collagen. There are fibroblasts and macrophages. And then the ground substance is filled with hyaluronic acid, dermatin sulfate, and chondroitin sulfate. If you remember way, way back at the beginning, these are three of our very classic aminoglycans, whose job, is to hold and maintain a bunch of that water, make sure to keep things hydrated. So there we go. All starting to come a little bit full circle. And the dermis is also made up of layers, just two though, instead of as many as the epidermis. We have the papillary layer. This is the more superficial of the two. It's made up of loose connective tissue. It also contains capillaries It has sensory nerve endings and these little structures called dermal papillae. These kind of extend up into the epidermis, kind of giving a good connection between the epidermis and the dermis, as well as providing additional surface area to transfer nutrients from the blood-rich dermis up into the epidermis. Next, the reticular layer. This is our dense irregular connective tissue. It has larger blood vessels, lymphatic vessels, nerve fibers, as well as some of our sweat glands, sebaceous glands, and hair follicles. Okay, a whole bunch of different types of cells that are involved within everything. We have keratinocytes. These are the majority of the cells in the epidermis, and they make keratin. That is the structural protein that hardens as it continues and goes up in order to strengthen the skin so that it can protect us. Next, we have Langerhans cells. These originate in the bone marrow. We stop when we think bone marrow. Okay, two things that are made in the bone marrow are either going to be our red blood cells or some type of immune cell. So Langerhans cells here, these are immune cells. They are antigen-presenting cells of the immune system. So what they do is they cruise around the skin, and if they find any foreign antigens deeper into the skin, they grab onto them and they take them to the lymph nodes, and they show the lymph node and say, hey, we've got trouble, and T lymphocytes are then activated in order to have an immune response. Next, we have melanocytes. Melanocytes, they produce melanin. We talked a little bit about melanin in our previous lecture when we were talking about the pituitary gland. This is the main area where we produce... Sorry, my mistake. We talked about melatonin. I believe I mentioned this when we were in that last lecture, how melanin and melatonin often get confused with one another as answer choices. So don't do what I just did. Make sure you get your rest and make sure you differentiate melatonin from melanin. Melatonin is for sleeping. Melanin is for the pigmentation of our skin. So melanocytes are derived from neural crest cells, and these transfer melanin to the keratinocytes and the stratum basale and the stratum spinosum. Here's a nice little chart giving you the cell types, the origin, primary role, as well as their location, just for easier studying. With melanin, we talk about skin pigmentation. There are 3 pigments that contribute to skin color. The first being that melanin, which is the only pigment that is made in the skin. So that's often the one that we consider and that we talk about. There are a couple others. Keratin, for example, which comes from plants that have beta carotene. Keratin, that's what we think. One main one that we think of when we think of beta carotene is carrots. Any kind of orange-ish colored fruit or vegetable has keratin in it, as well as some not orange. Some of our reds and yellow vegetables and fruits also have keratin in it. But carrots, sweet potatoes, those are the main ones that we think about that have the highest concentration of keratin. And it is true that if you eat carrots or keratin-rich foods every single day, you will actually be slightly tanner. If you eat too much of them, you can actually get a condition called hyper-beta-kerotonemia, which means that you have lots and lots and lots of excess beta-carotene in your system, and that can actually turn you an orange-yellow color, much like jaundice does with the liver, except when we have liver dysfunction, we can also turn yellow because the liver is not pulling out all the yucky stuff that it needs to. Main difference between those two things is look at the eye color. With the liver, the eyes are going to be yellow as well. The whole whites of the eye, the sclera, is going to be yellow. And with beta-carotenemia, it's just going to be the skin. Next is hemoglobin. If we have lots of hemoglobin, we can be a little bit more red, a little bit more flushed, where if we have low hemoglobin, we tend to be a little more pale. It's like people with anemia who have less red blood cells and less hemoglobin, they tend to be paler in color. The blood supply to the skin. So there are arterial and venous plexuses in two areas between the papillary and the reticular layers of the dermis, and then between the reticular layer of the dermis and the hypodermis. These also contain arteriovenous shunts. Think of these as like little bypass valves. So that way they can bypass the superficial capillaries whenever we need to conserve heat and or increase blood pressure. So the skin is a really, really large organ, and it can dissipate a lot of heat. So if we're ever in a very cold situation, what actually happens is the body shuts off the blood flow to the top layers of the skin, so that way we don't lose as much body heat in order to make sure that we maintain body heat in our core. So that is the purpose of the arteriovenous shunts. Here's that circulation. Then we also have nerve endings. Nerve endings, you're going to talk a lot about these when you get into more of your neuro courses, but I want to bring something, let you know, talk about something very specifically with these, and something that you can start doing now to fine-tune your palpation skills, your light touch. Because really that's what a lot of chiropractic is we need to be able to feel the things below all of the different tissues. So I'm going to give you a couple of hints of things you can start doing, especially if you're in the blended program. If you are a blended or online student, start doing these things to practice. So I'll get to those in a sec. There are two types of nerve endings. There are encapsulated, which means that they're covered by connective tissue, and then there are free nerve endings. So these are free, these are open, they're not covered by anything. We have, if the encapsulated ones, we have Meissner's corpuscles. These are located in the papillary dermis, specifically in areas of high tactile sensitivity, the fingertips being the main ones, the lips and also the palms. They detect light touch as well as low frequency vibrations. So these are the type of nerve endings that we actually want to train as chiropractors. We want to be able to use a very light touch, because you actually feel more when you touch something very softly than when you press really hard and aggressive. So two things that you can do. I think I have one here. Okay. I have a quarter here with me, but what you can do is take a bunch of, actually I have one right here. I have a penny. Take a bunch of pennies and dimes and put them into a bag. And what I want you to try and do is reach into the bag and very, very gently feel the penny and the dime. Feel each one. Feel the front. Do this with each finger. Try and, without looking at it, identify, is this the front of the penny or is this the back of the penny? What type of penny is this? Is this a shield penny like this one is? I'm not sure if you can see, it has the little shield on it. Or is it the Lincoln Memorial penny? Which side has Lincoln's face on it? Is it a penny? Is it a dime? What is it? Try and start to feel those very, very small differences between the two. This is going to train this light touch to be more and more sensitive. The other thing that you can also do is get yourself a dry spine. I don't think I actually have my dry spine anymore. I do have my full skeleton somewhere. There it is. So get yourself a spine model of some kind. I can't get it out. My wife put it down underneath my desk and then moved some stuff, so it's stuck down there. But what you're gonna do is lay out your spine model on on the couch or on your table or something like that. And you're going to close your eyes, and I want you to start at the top and feel each individual vertebrae as you move your way down. Get used to, how much space is in between each vertebrae in the cervical spine, in the thoracic spine, in the lumbar spine. And what you can start to do is put layers of stuff over the top of it. Start with a single layer of a sheet. So put a bed sheet over it, and feel and be able to count down. So have somebody tell you or say a specific vertebrae, so like say T8. And then starting at the top or from the bottom, close your eyes and try and palpate and feel and count up to T8 or down to T8, however you want to do it. And then take off the sheet, count, and see if you were right. If not, keep working on it. Keep doing it with a sheet. And then after you feel pretty comfortable with that, fold that sheet in half. And then fold it in half again. And then go get a towel. Get a thicker towel or get a thicker blanket. And keep on adding layers as you do this. This will really help fine-tune your light touch and make it much nicer when you start to see patients and palpate actual patients instead of having really heavy hands. Sometimes I make the mistake of palpating a little more forcefully than is needed, and especially in a certain patient demographic, women, children, geriatric population, it can be very sensitive. So you have to make sure that you scale back the amount of force that you have in order to be able to make sure that they are comfortable. Okay, back to the material. Meissner's corpuscles, they're in the papillary dermis, and they do the light touch. Pacinian corpuscles, these are in the deep dermis or the hypodermis, and they are deep pressure and high frequency vibrations. Finally, the free nerve endings, these are found throughout all of it, but they are especially abundant around the hair follicles, and these detect fine touch, temperatures of hot and cold, and then pain, which is called nociception. These are our Meissner's corpuscles. There's one right here. You can see another one right here, little capsules. These are our Pacinian corpuscles. You can see the little capsule running around it. We have our free nerve endings. These really just come up into everything. It's a couple additional structures. We have our sweat glands. Sweat glands are also known as eccrine glands, and these are pretty much everywhere on the entire body, with the highest concentration on the palms, the soles of the feet, and the forehead. They are simple, coiled, tubular glands. Think way back, our fourth lecture was on the epithelium, and we talked about the coiled tubes, right? Where it looks like these little coils, where in the bottom we have secretory cells, and then we have the duct lined with cuboidal cells, or stratified cuboidal cells. So the secretory portion at the very bottom that makes the sweat, that is simple cuboidal epithelium, and then the rest of the tube coming up, that is stratified cuboidal epithelium. And it does what's called merocrine secretion. Merocrine secretion means that it's released by exocytosis. So essentially we form the sweat components all inside. We wrap them up in a nice little ball called a vesicle. We push it towards the edge of the cell, the top of the cell, and then it merges with the plasma membrane and releases the sweat out into the sweat gland and through the sweat duct. So those are our sweat glands. A nice little picture of some of those ducts right there. There's the secretory portion. Additionally, we have the hair. It's found in the thin skin. It grows from the hair follicles, which we say they originate up here in the epidermis, and they extend all the way down through the dermis, sometimes even into the hypodermis. Don't worry too much about this. This is just a little extra info, where the hair cells are actually composed of keratin, and they have a couple of different parts, but that's that's just some extra information I included in there. Sebaceous glands. These are similar to sweat glands, but they're slightly different function. Sweat glands are for helping to thermoregulate us. Sebaceous glands produce a substance called sebum, which is an oily substance meant for lubricating our hair, waterproofing, as well as having antimicrobial properties. my cat has decided that the cords on the back of my computer screens are fun to chew on. So it's a constant battle of keeping her out of there. Okay, so we produce the sebum, and it is produced through what is called holocrine secretion. This is where we literally get parts of the cells disintegrating in order to release this oil. The sebaceous glands are often found right next to the hair, the hairs and the hair follicles. Just like right here, these are our sebaceous glands, and this is the ducts running to them, and then it goes and it travels up out of the hair follicle itself. Finally, we have a muscle called the erector pili muscle. These are smooth muscles attached to the hair follicle base as well as the dermis. When we have a sympathetic nervous system activation, like that fight or flight response, these cells will contract. It causes our hair to stand up on end. That's that goosebump feeling. This is thought to be kind of a reaction meant to, like in our animal counterparts, where our hair would stand up on end and make us appear larger or more threatening than we actually are. But because we don't have very much hair on our bodies, we just get that goosebump sensation. Here is just another little chart, just kind of generally outlining each of those different layers, all that kind of good stuff. Just a little study help if you want to use it. And that is it for the Integumentary System. Thank you guys so much for watching this video. Of course, leave a like, subscribe if you haven't. Comment any questions that you might have. Understand, just keep pushing through. We're getting close to the end of this specific course. So as always, have a wonderful day and good luck studying.